Some legends are told, sometimes the dust turns to gold, but you will remember me. All right, have you ever wanted to learn microbiology? Well, you're gonna, whether you like it or not, you're gonna. Now today's lesson is gonna be over chapter 3, okay? If you haven't learned chapter 1 or 2, I'm sorry, that's on you. That's on you. Prokaryote cells and eukaryote cells. Now, what's the difference? You know, prokaryote, think bacteria and all that tiny stuff. Eukaryote, think us, think you, think the food we eat. Yeah, pretty much anything that's a tree. Doesn't matter, you know the difference. Now, let's learn why they're different. Prokaryote cells, they don't got a nucleus, dog. Their DNA is just out there. There's no membrane holding the DNA or RNA, depending on what type it is. But that doesn't matter either. They have no organelles, they divide by binary fusion, and they have no histones. We'll talk about histones later. Now, eukaryote cells, they have a nucleus, they have organelles, they divide by mitosis, which is replicating the DNA. You know mitosis. And they have a polysaccharide, membrane. That's what, like, the outside is made of polysaccharides. Prokaryotes have a peptoglycan membrane. Different molecules. But we'll talk about that. Talk about it. You'll learn. Now, we're going to discuss prokaryote cells and their weird oblong shapes. They can be a pill. They can be a big old oval. They can be a sphere, twisty. They can be a rectangle. They can be whatever they want, really. Depends on the bacterial cell. Sometimes I don't feel like being in a square, you know. I might choose to be a circle. So that's just how they act too, kind of. Now, what are the names? When we're looking in a microscope, thinking, wow, that's a lot of cells. Some of them come in pairs, some of them come in clusters. You know, they like to huddle up. They're like, come here. Pairs, Diplo. Think Diplo, you know, the singer, drop banger songs like When I Met You in the Summer. We fell in love as the leaves turn brown. Anyways, and then we have chains, you know, staph infections, staphylococcus. Those come in like big old little chains. You don't really need to know this, just think blah, blah, blah. Now, we're still focused on the prokaryote cells, obviously. You know, eukaryote cells, we know about eukaryote cells. Prokaryote cells have an external wall called the glycosacs, and you don't even know the name. I don't even know the names. I took anatomy, couldn't pronounce half the words. This tendon, that tendon, something. We got the quads, that's all I know. They are viscous and gelatinous and polysaccharides or polypeptides. That's what they're made of. Now, they help them firmly attach to things. You know, they like to grab on bacteria. They're on me, dog. They're on you, dog. They're on you. This helps them sow phagocytosis. Yeah, phagocytosis. You know, they don't want to be eaten. They just don't want to be eaten, dog. I wouldn't want to be eaten either. That's why I'm not a bacterial cell. Flagellus. Now, how do these guys move? Are they like wiggling around? Kinda. But they have a little tail that whip. Think sperm cell, you know. How'd that guy swim? How'd I get here? How'd I win? Well, I had a little tail. And that tail helps make them with motility. Motor functions. Like automobile, motility, moves. Easy peasy, lemon squeezy. And there's 3 parts. They got the hook. That's on that hooks them into the inside of the cell. They're there. The flagella is strong in there. Then we got the basal body, another very key part because it consists of rods and pairs of rings. But it depends on if it's a gram-negative or a gram-positive cell. We'll talk about that too. You'll learn, dog. The flagella allows them to rotate, tumble. They're made up of proteins and have different types of antigens. Antigens, you also probably need to know that, dog. I'm sorry. If you're interested in this topic, you gotta know what an antigen is. It's basically just like something that tells another microorganism, hey, I got this. It's like a communication in many different ways. Even in our bodies, we have antigens and hormones and a bunch of different things. And then the fimbriae and pili. This is on the outside, again, membrane. We're gonna break it down. It is all on the outside. Fimbriae are like little tiny arms, like little grabbers. They just out there touching you. They're touching me. You're gonna get touched. All right, now pilis, they also help in mobility. Pili, I don't know. They kind of like help in mobility, and then they also help like, come here, brother. What's up, brother? They transfer DNA to other cells. So like they're like little kind of like, prepare to get boarded. Oh no, they've caught us in the, what's that Star Wars, like when the Death Star, when the Millennium Falcon got caught in the Death Star? Tractor beam, it's like that. And then like when the ship's board, yeah, it's like that. Just think that. Just think tractor beam with pili. Doesn't matter. Pili are typically longer and fewer in number than fimbriae. That's because, they're found virtually in all gram-negative bacteria and not many gram-positive. I don't really know the difference yet, fully. Besides, like, what the difference is. Like, doesn't matter. Let's go to the cell wall now. The cell wall, very important, dog. It's not present in all, actually. It's, I think it's also gram-positive, maybe. Don't quote me on that. Prevents osmotic lysis and protects the cell wall. Think like Attack on Titan, Wall Rose, Wall Maria, out there. Everything else outside the cell wall, those are Titans. You're getting eaten. You are screwed. There's no doubt about it. These are made of peptoglycan in bacteria, not in eukaryote, so plant cells, this is different. They contribute, they can cause disease. If this thing touches you, dead. RP to Arthur from Red Dead 2, he died from TB, man. I think that's a virus. Never mind. Peptoglycan polymers can be N-acetylglycosamine. Yeah, N-acetylglycosamine or N-acetylmuric acid. Nag and NAM. She nags on my NAM till I peptide glycan. She nags on my NAM till I peptoglycan. That's how I remember it. That's literally how I've been talking to myself. Like, how do I remember nag and NAM? How are they? They're both peptoglycans. So she nags on my NAM until I peptide. And yeah, these are rows of polypeptides linked together. Hydrophilic on top, hydrophobic on the bottom. So they have tails. I hate molecular structure, so I'm not going to even show you an image of it because I don't care to look at the image of it either. Finally, I was talking about these gram-positive stuff. Like you're thinking, what the hell is gram-positive? Don't know. I'm going to tell you what I do know. Gram-positive cell walls, thick. These boys are thick peptoglycan. So gram-positive, thick, positively. And Tyric acids, I don't know, they got acids in them. Lipoteic acid links cells wall to plasma membrane. So whatever the butyric acid and lipo that it binds to the cell wall. Or like the plasma membrane binds to the cell wall. Whatever that acid is, it does it well. They carry a negative charge, regulate the movement of cations, and they are polysaccharides. They provide antigen specificity. I cannot speak. Specificity. Say that 5 times fast. So basically that just like means that they are the reason some cells are like they can do what they can, because they're very specific. If that cell wall has the antigen polysaccharide, they're getting in you, dog. They're on that food. They're gonna mess you up. That KFC, if a cell wall, if that bacteria was on there and it had like a certain antigen and the gram-positive cell wall was there, I'm sorry, you're gonna be out. You're gonna have to see the doctor and be like, hey bro, I need those steroids. I need that. I need it. That was terrible. Let's go to gram-negative cell walls. These are thin, negative, that's unfortunate. Thin polyglycans. Outer membrane is made of polysaccharides, lipoproteins, and phospholipids. It's kind of familiar, kind of similar to eukaryote cells. Look at that. This one protects against phagocytosis and complements, antibiotics. Phagocytosis and antibiotics. It just bounces off. Ain't happening, brother. Ain't happening. This is made of lipopolysaccharides. So like the other one, didn't have. They had acids. They had that lipo-TH acid. This one's lipoproteins, lipopolysaccharides, LPS. O polysaccharide functions as antigen, like E. coli. And then lipid A is an endotoxin embedded in the top layer. So this one's still getting you, dog. This one's still gonna mess you up because of the lipopolysaccharide, LPS, lipopolysaccharide. These are terms you gotta remember. I have to remember too, actually. And it has porins. So these are tubes, proteins. Porins are proteins that help transport their little channels, so the little holes, porins, pores, space. You know what I'm talking about. Cell walls and the gram stain mechanism. Don't care. We're gonna skip this. Literally do not care. Study on your own. Learn on your own. But you're saying, why? You're supposed to be teaching you. But I say to you, wouldn't you like to know, Wither Boy? Wouldn't you like to know? Because I'm not gonna tell you. I'm not gonna tell you. Now, atypical cell walls. They're acid-fast cell walls, like gram-positive cell walls. They're waxy, mycelic acid bound to peptoglycan. I'm saying these words properly. Are you? You don't even know if I'm saying them properly. Can you trust me? Can you trust me? Trust me, please. Microbacterium, no, I don't care about that either. Plasma, cytoplasmic membrane. Phospholipid bilayer that encloses the cytoplasm. That one's actually very important. This one, the plasma membrane, very important. There's a star next to it. I remember her talking. Yeah, gotta remember this. Whatever this is, remember it. Peripheral proteins in the membrane surface. So remember the porins? They're here. Plasma membrane. They're letting in your buddy, but they're semi-permeable. Now, I had a professor last year that was very good about, hey bro, this membrane is semi-permeable. Think like you open the door for your, you're in college, I don't know, people are coming to your door. I don't know why people are coming to the door, but she was like, there's this one person you don't really like, but you're chill with, they brought food. Come on in. Obviously, you're gonna let your friend in. You're gonna let your girl in. They can come through the membrane, selectively permeable. Now, say this person is really annoying, you don't really like them. They knock on the door and they're like, hey, can I come in? Looks cool in there. You're like, nah, bro. Nah. What are you doing here, bro? Why are you here? Get out. That's what semi-permanent are doing. That's why they have the proteins and whatnot to help them be like, no, 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 no. Do you have a wristband? Do you have a wristband? Think that. Now the functions of the plasma membrane are, again, selective permeability, like I talked, enzymes for ATP production, acetyl triphosphate, acetylcholine, I don't know what ATP stands for. Forgot. Look at me. I guess it makes ATP. We need ATP. You know what it is: glycolysis in the mitochondria, powerhouse of the cell. ATP. They don't have mitochondria though. They have ribosomes. So they also, some of these plasma membranes have photosynthetic pigments that help. These are chromatophores. Yeah, chromatophores. That's a little pigment, you know, that helps out. They should be like, hey, bro, I need the sun to help me ATP. Make me live, son. Go get some sun tomorrow. Damage to the membrane by alcohols, detergents, antibiotics causes leakage of cell contents. I'm leaking all my cell contents because of the antibiotics. Plasma membrane should be doing that, dog. Now, I was talking about how things get past the membrane. There's different functions to that. The movement of materials is very important. We got passive, we got active. Passively, I could be thinking about a lot of other things like maximum pulse, or like I'm breathing. I just remember I'm breathing. I'm gonna be actively doing that for a minute. So are you. Remember to breathe. You're breathing right now. Sorry. Basically, passive means no energy. You know, substance can move from high concentration to the low concentration with no energy needed. Yay. You eat a piece of meat or food, it's done. You're letting your body take care of the rest. Technically energy is involved, but the example is like you're not doing it. You're like, no, you're not. No. It's done. It's passively going through you now. Active process is like, oof, I gotta walk to class, go upstairs. I gotta work out, you know, active. So this is energy needed to go from walking upstairs, you know, low to high. You're gonna need energy to walk up those stairs. You're going from low to high. Walking downstairs is easy. Also, that can help you remember. That would help me remember. Walking downstairs is easy, so you're going from high to low. Now, there's different types of passive. There's simple diffusion, which is like osmosis and a bunch of other things. Looking at my notes. And simple diffusion is the movement of a solute from an area of high concentration to area low. Again, you're walking down the stairs, you're just throwing stuff. The solution is just it's riding down the hill on a snowy day. Did you ride down the hill on a snowy day? You should have. Now, facilitate diffusion, man, I need a car. I need to get there somehow. So there's proteins, transporter proteins in the membrane that help you get there. They're like the MARTA in Atlanta. Just don't take the MARTA. And there's specific and non-specific. You know, transports ions and large molecules across the membrane with the concentration gradient. Concentration gradient? What's that, brother? I'll tell you about it. We have isotonic, hypotonic, and hypertonic. Those are the concentrations. Isotonic, we're even. You don't owe me nothing. This water, it's not isotonic, I don't think. But there's nothing different on the outside, nothing different on the inside. Hypotonic, that means the concentration outside, very, very low. I'm bloated. And so you might need to throw up, you know, you're kind of like stuffed up. Gotta let some of it out if you were a cell in a hypotonic solution. Now, if you're in a hypertonic solution, more than likely you're like, oh, I need some water. Oh. Hyper, think too much. You know, some kids are hyper, you have to give them some sugar. I'm hyper after you give some sugar. I'm hyper right now. So obviously the solution around you has more than what you have. So you want to get to equilibrium and take some of that in. So water. Oh, you should also know the names of this. So in hypertonic solutions, plasmolysis. That's what's happening to the cell. Plasmolysis. Look at that. Hypotonic solutions, if the cell's in a hypotonic solution. Osmotic lysis. Isotonic doesn't have one because it's already at equilibrium. So there you go, man. Osmosis Jones. What did he do? Did you watch that movie? You should watch it right now. Just to say you studied. You can watch a movie and say you studied. Just water moving across the membrane. You don't even know that. You do need to know that. But like, come on now, man. Now, active transport requires a transporter protein, and it requires ATP. Again, it's active. It goes against the gradient, so more than likely this is probably a hypertonic solution. It's like, you know, I still need that protein, I still need whatever's outside, but I don't want everything coming in. So there's group translocation, whatever that is. You got a big group? Let's go, let's all go. This requires a transporter protein and phosphenolpyruvaric acid, PEP. I love me some little abbreviations, PEP. Phosphenolpyruvaric acids until transporter protein? That doesn't make any sense. The substance is altered as it comes across the membrane. Now, how does it do that? I don't really know. I just know that I wrote it down. That's good enough for you too. Cytoplasm. Let's get inside the cell. We're fun. We're finally getting up in there, you know? Getting up in there. This is what's the jelly. It's the jelly filling. It's like the inside. It's all the stuff that holds the cell together. It's 80% water. Remember that. Maybe. Plus proteins, carbs, and lipids and ions. Probably like less. That's probably the other 20%. It's the cytoskeleton. Cytoskeleton is a structural kind of jelly. Like it's a little harder. It's a little harder, dog. It's the skeleton of the cytoplasm. There's some anatomy to that, but I don't think you have to know that. So go you. Proud of you. Give me a high five. Did you have a good day? Okay, nucleoid. It's the bacterial chromosome. It's circular. It's just like a circle. Think like Tony Stark. He just got the Mobius strand. Mobius, whatever it was called. He was like, I just saw time travel. It was a circle that somehow folded in on itself, and up was down and down was up. I don't know why I explained it. It's just a fucking circle. It's just a circle of DNA, literally. Genetic information. Now, plasmids are extra chromosomal genetic elements, and they carry non-crucial genes. So these are like the little notes, like it's the notes app for the cell. Literally, plasmids. It's the notes app. It's like, hey, I got hit with the antibacteria last week, you know. Make note of that, Siri. And she did. The plasmid did.
Uh-huh.
Come on, Siri. Don't do that. Shh. Yeah, it's a plasmid. You should know that. We should actually go back to the histones. They don't have histones, and histones in a eukaryote cell would protect the DNA. That's what makes a chromosome. A histone is the protein like around the DNA, which makes that little. You don't even really know this, because I don't think I need to know it. Ribosomes. That's the site of protein synthesis. I'm thinking like, how is all this stuff made? Proteins are like key to the whole everything. They literally read the DNA and all that. That's ribosomes, made of protein and ribosomal RNA. Now, RNA is a little different from DNA because it's like the stuff that's been transcribed, and it's like the, hey, do this for me, because I said so. Because it was like, that's what's in me, the DNA. Anyways, it's made up of 50 subunits plus 30 subunits equals a complete 70S ribosome. It's like a little plate on the bottom that kind of is like, I think of it as like a conveyor belt. So you got the little 30 subunits on a conveyor belt, and the top is like what's reading it. I don't think this is how it actually works, but when you see the picture, maybe right here. There might be pictures all throughout this. Who knows? And then endospores. Yeah, endospores. This is when the cell is like gone. They're broke. They're broke. They're broke cells pretty much. They don't got enough money for food. They lost their house, and like they were like, I got that child support in a month. What am I gonna do? They go into endospores. This is a resting cell and produced when nutrients are depleted. It's resistant to heat, chemicals, and radiation. Disinfection, yeah, none of this happening. Produced by Bacillus and Colostrum. Sporulation, endospore formation. Germination, endospore returns to vegetative state. Germination, it's back. It's been germinated. I think also like the production, isn't that what like happens to sperm and egg? It's germinated. Germination, I think. Don't quote me on that. And then the flagella and cilia, we're back at that. Both consist of microtubules made of proteins and tubulin. So tubulin is a protein that helps with the microtubules in the flagella and cilia. Now that's all I have written down in my notes. Is there anything we'd like to cover, class? Glad you asked. What do I skip? What do I need to talk about? What do you need to talk about? What do you need to learn? Maybe you should learn chapter 1 and chapter 2. We're not going to do that right now. Cell wall, the glycosacs, the flagella, talked about that. Membrane, talked about that. Cell negative, gram cell, yeah. Yeah. It's basically it. Oh boy, I hope I didn't learn microbiology today. I said microbiologically. I hope y'all watch those. I truly do. Those little cat videos where like it's like evil Larry. And evil Larry does something very evil, like turns you into a sandwich or touches you. It's evil Larry, it's not me. Lipopeptoglycan polymers. Remember, she nags at my name until I peptoglycan. I remembered it. Did you? That is about it. Prokaryote, eukaryote. I don't know why we didn't even get to eukaryote, but if you don't have to know it, you don't have to know it. Isn't that a blessing for when you're learning? Maybe. Let's see. Let's go ahead a little bit. Cell wall and glycocalyx. Glycocalyx. The cell wall is found in plant, algae, and fungi, made of carbohydrates, cellulose, plants, chitin. I'm going to say chitin in fungi, and glucan and mannan in yeast. The glycosacs, the glycocalyx, is a carbohydrate bonded to proteins and lipids and plasma membranes. It's found in animal cells. We got that. And I guess so do bacteria? Are we bacteria? So a lot of existential thoughts right there. How tiny are we in the universe? I don't know, I should really need to know. I don't even care to know. But I think that's everything. I think that's everything you need to know. Me. This is for me too, if you didn't get that. Osmotic pressure, osmosis, blah, 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 passive transport facilitated diffusion. Talked about that. You don't gotta be here no more. I'm just reviewing everything real quick. Simple diffusion, talked about that. Movement of solid from an area of high concentration to low. Simple. The high concentrations outside, the low concentrations inside. Sometimes. You can't have a high concentration inside and then you get rid of it simply, passively. Active process, talked about that. Do we need to know back the shapes of the cell? Bacillus, rod-shaped, caucus, spherical shape, spiral, fibro, spiralium, spear, chip. That's spiral. Star-shaped, I didn't even tell you about the star-shaped one if you're still here. I did tell you about the rectangular one, so that's good. I was kind of there. I was kind of in that. Not going over the names. Just think pairs, clusters, chains, groups of 4 is tetrads, and cube-like are saccharinae. Glycosacs, external wall. 2 types, capsule and slime layer. I don't think I told you about that, but you don't really need to know that. I don't think I need to know that. Flagella, 3 parts. Oh my god, I forgot to tell you about the filament. It's the outermost, it's the dangly thing. That is the tail. I told you about the hook and the basal body, but I didn't tell you about the filament. I'm so sorry. I apologize. Please accept that apology. Now, the basal body consists of a rod, a pair. I already told you about that. And it's hooked into the cell wall, so it's not going nowhere. And it does that differently in gram-positive and gram-negative cells. You have to look that up, maybe, depending on if you want to. The taxis. The flagella allows bacteria to move towards or away from stimuli. So, if it feels something down there in the microscopic world, it's like, hey, Brian, get me the heck out. Get me out of here, dog. Taxi! Taxi! Oh shit, I got a taxi on me. It's gonna fling itself around. Rotates and tumbles. Membrane, hair-like, pili. I already talked about that. Peptoglycan. Cell wall again. Sheet peptides on my glycan. The cell wall also contributes to pathogenicity. That's why we get infected. Cell wall. Just know that. We have to know why we get sick and why these are pathogenic. It's a very important topic. You should know that. Composition and characteristics. Where did I write that? Composition, and did I skip that? No, I didn't. Maybe I did. Probably did. No, I didn't. I just didn't write the header. Sorry. I'm struggling with myself. Rows are linked. Talked about that. Gram-positive. I talked about how it's thick. And teostric acids. Lipoteostric acids. Talked about that. Then peptoglycan on the gram-negative. That's unfortunate. Protects from phagocytosis, complement, and antibiotics. Made of lipopolysaccharides on the gram-negative. LPS. Polysaccharides, yeah. I'm just going over everything I already talked about. Just rewatch the video. I hope I explained very well, I hope you learned something today. If not, Larry's coming for you. I'm sorry. Larry is gonna come for you. It's evil Larry too. It's not normal Larry. Evil Larry is gonna do something evilly. It's just how it be.